This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 94, recorded on December 18th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, howdy. How are you doing?
I'm well, Elio. Good to hear from you again.
Yes,
Elio, I introduced you from Small Things Considered. Do you like that, or would you prefer something else?
Sure. Well, I have too many affiliations. I have an appointment, an adjunct appointment at San Diego State University and the University of California, San Diego, and I'm emeritus from Tufts University. So that's too many things. So, yeah, Small Things Considered is what my identity
Yeah, you spend most of your time there, right?
It's
Absolutely.
sort of like introducing the president of the United States. When he walks into a room, they just say, the president. They don't list his CV. And I don't think we need to list Elio's CV. I mean, Small Things Considered speaks volumes.
He was the president of ASM.
This is true, so you can introduce him as the president.
Yeah.
Past.
Well, you know, but
you don't want that job.
Don't
again.
they address former U.S. presidents as Mr. President still?
They still do.
Oh.
So you can just introduce him as the president.
Okay. By the way, also joining us from the Medical University of South Carolina is Michael Schmidt.
Hello, everyone.
How are you?
I'm doing well today.
Are you heading out to Chicago for the holidays?
Yeah, I'm going to go visit my father.
That's good. Now, what is he, 95?
He's 90. He's a radio station. He's 93.5.
Wow.
Well, once you reach the radio station phase in your life, you can do whatever you want.
That's pretty impressive.
Yeah.
I heard of that.
Yeah, he's a veteran of the Second World War. He survived the Battle of the Coral Sea and has all sorts of stories about kamikaze attacks and zeros flying on the out of the sun. And
Michael, you ought to sit down with him and record a conversation, because that stuff, there aren't many people left who went through that.
And he was on the USS Lexington, an aircraft carrier, and he was a lookout because, unlike his son, he had superior vision. And so he was a lookout up in the Pry Fly. And so he actually got a presidential citation for spotting this one kamikaze attack and actually saved literally the carrier and the fleet.
Nice.
Yeah.
Well, have fun out there.
Oh, yeah. I'll be shoveling snow because the weatherman says it's going to be snowing on Christmas Eve in Chicago. So I'll get to reacquaint myself with a snow shovel, which is never pleasant.
That's right. Well, today we have a couple of papers for you. But before we start, I just want to say this is the last episode of TWiM for 2014. The next one you'll hear will be the first week of January. We did 23 episodes this year.
That's a lot.
What do you think?
I think we did well.
Elio, you happy with that?
Yeah, it's quite a pace. I must say, this is, just in case people wonder, this is work. It's pleasure, but it's also work.
Well, plus you didn't count the ICAC lives that I did for ASM at the Inner Science Conference on Antimicrobial Agents and Chemotherapy. Yep. I did 5 ICAC lives, which they're available on the web, so you can go off and see them. And we talked about all sorts of interesting things like antibiotic stewardship and carbapenemase-resistant Enterobacteriaceae and making hybrid bugs. So it was a pretty interesting session at ICAC too.
Yep. So we look forward to more in 2015. More more TWiMs, and more all sorts of other things. So let's get down to the science now. Elio, what do you have for us?
Okay, well, I have a couple of papers that deal with symbionts that carry out nitrogen fixation. And, nitrogen fixation is really one of those giant subjects that make the world go round. And I think that we have barely touched on it in TWiM, so I'll give a little bit of an introduction, if you don't mind. So nitrogen fixation refers to the fact that the source of the source of utilizable nitrogen for living things is fixed nitrogen that becomes something other than dinitrogen atom molecules. This means it becomes essentially glutamate. And when it becomes glutamate, which is an amino acid, of course, which has nitrogen, it transfers that ammonium group to other amino acids, or in the making of other amino acids, and they become proteins and so forth. And the same thing goes for nucleotides. So nitrogen is essential for life, and we cannot do much with nitrogen. We breathe nitrogen galore. 80% of what we breathe is nitrogen, and it doesn't do us any good. It comes in our lungs and out our lungs. So how does nitrogen get fixed? Well, it is done by prokaryotes only. And there's no known eukaryote that I know of that can carry out nitrogen fixation. But some bacteria, and some archaea do.
So, Elio, if you get rid of all the prokaryotes, we would have no fixed nitrogen and we would all die, right?
Okay, well, that brings me to how we dealt with this question.
I just wonder if that's true or not.
Are you going to talk about the Haber process?
You can.
Yeah, eventually, sure.
Eventually.
But right now, let me say that we asked this question: what would happen in the world if all bacteria and archaea went on strike? And the answer, which was contained in a Talmudic question, and it was answered by David Lipson, a professor at San Diego State. He did a calculation and figured out that the amount of available nitrogen by natural sources as opposed to artificially made nitrates, fertilizers, would be sufficient to sustain life for maybe a year. And that after a year, we would depend entirely on making nitrogen for plants from the process that Michael mentioned, the Haber process, which is a way of using energy galore for fixing nitrogen. So the Talmudic question was answered, and recently, just today, an article appeared in PLOS, and the reference of that will be in the show notes, which mentions David Lipson's calculation and makes note of this particular Talmudic question.
Elio, what's a Talmudic question?
I thought you'd never asked. Okay. In our blog, we have a series which we call by that name, and which simply consists of questions which are derived from the old idea that the Talmud, the commentary of the Bible, was studied in the old days in a way that where the question was often the point and not the answer. In other words, you don't really have an answer necessarily. But the issue that it brings up, that the question brings up, are interesting. So the question in this case was, what would happen if bacteria and archaea stopped working? And the answer is a guesswork. It's obvious, but it brings up a number of issues, and that's what we enjoyed. And in this paper in PLOS Bio, they mentioned this particular paper, so I'm very happy with it. There's another Talmudic question which we asked. Why? So, I'm sure that most of our listeners have heard of root nodules in legumes and other plants, which are made up of bacteria which fix nitrogen and make it available to the plant directly. so that if you grow peas or alfalfa or things like that, you don't really need as much fertilizer, if any, at least not nitrogen-containing fertilizer, because the bacteria do it for you. Now, these bacteria, let's just deal with that for just 1 second. There are going to be a number of digressions today, so I hope the listeners don't mind that. We do it all the time, anyhow. In root nodules, the bacteria are sort of terminally differentiated. That is, they become able to fix nitrogen and stop at that point. That is, they appear to be either dead or close to dying, although they're metabolically very active. So the question became, why didn't these guys become organelles? Just like bacteria became mitochondria for the sake of making energy, and chloroplasts for the sake of capturing light, photosynthesis, why didn't nitrogen-fixing bacteria become an organelle? You could call it a nitrochondrion or an azoplast, okay? Call it whatever you want.
I like mitochondria.
That's great. I love it.
Nitrochondrion, okay. But there are no mitochondrions.
This is
maybe
true.
It's happening in front of our eyes. Maybe evolution is taking care of that. But it makes good sense that the plant cells ought to have organelles that can fix nitrogen instead of just having to have a whole bacterium carrying a whole bacterium alone, which is a bigger load. Anyhow, it hasn't happened yet. So look out for nitrochondria. By the way,
you better copyright that word before Craig Vettner's company does.
Okay. Well, you heard it here. Anyhow, nitrogen fixing by bacteria and archaea is not limited to nodules. In fact, the bulk of it is made by free-living bacteria like Azorobacter, which carries out a lot of it. I want to give you some numbers. On land alone, the terrestrial production of fixed nitrogen is between around 100 million tons a year. Numbers vary a little bit. It's less clear how much there is in the ocean, but it's probably the same order of magnitude, something like that. The amount of, so let's say 100 is sort of a round figure, 100 million tons. The amount of fertilizer which is made by the Huber process and the likes is about 80 million. So there is about the same amount, close to the same amount is made by people, by man, and industrially, as it is made naturally. However, there are other sources of nitrogen, and one of them, which is interesting, is lightning.
Yes.
It's unsuspected. Lightning causes nitrogen to become ammonia. Maybe I didn't say that the first thing that happens to nitrogen is it becomes ammonia and is taken up is used by the bacteria that make it.
And that's the point you should make, is that the nitrogen used in biology is in its most reduced form as NH4+.
Right, that's right.
So
the process. Go ahead.
The Haber process that you mentioned is important because that's a way to make ammonia from gaseous nitrogen, and that allows fertilization, right?
Yeah, nitrate mainly.
Yeah. Otherwise, we wouldn't be
ammonia as well. Yeah.
Otherwise, we wouldn't be able to feed the world's population.
That's correct.
Okay,
that's correct. So it's a giant process. Now let's look at it. It's a very interesting biochemical process because fixing nitrogen is energetically very expensive. The reaction, which was discovered, or nitrogen fixation was discovered in 1901 by Bayering and somebody else, consists of taking a nitrogen molecule, dinitrogen, and which is a triple bond. That tells you how tough it is to break it up, with 8 hydrogens and 8 electrons, 8 protons and 8 electrons, and that gives you 2 molecules of ammonia and a molecule of hydrogen. It requires 16 ATP molecules, so it's expensive. And that biology has learned how to do it is interesting. It's really a different kind of biochemistry. The enzyme involved is called nitrogenase, and it is pretty much the only enzyme of its sort. That is, all nitrogen fixation is done by nitrogenases. Of course,
and they're killed, inactivated, or whichever adjective you want to use with it, by molecular oxygen. So this is an old enzyme to biology of our planet, because if Earth had oxygen before life emerged, we would have never emerged, because there would have been no way to take the noble gas nitrogen and use it for protein. We would have evolved some other mechanism to make our structural molecules
you
from.
couldn't have proteins or nucleic acids or anything like that. That's correct. And the strategy, let's stay with that for a moment, because it's interesting. The strategy that organisms use to protect the nitrogenase is varied. Okay, so you have to protect it somehow. And so some of these bacteria are simply anaerobes. They don't live in the presence of oxygen, and so oxygen is not a threat. Others make specialized structures, like certain cyanobacteria, which are filamentous, that are rather the chains of cells. Every so often, every 10 cell or so is differentiated in a cell called a heterocyst. And heterocysts do not carry out photosynthesis. In other words, they don't produce. Cyanobacteria, by the way, are known for their oxygenic photosynthesis. That is, they make, in the process of photosynthesis, they produce oxygen, and that is thought to be the source of oxygen, originally the source of oxygen in our atmosphere. But heterocysts don't photosynthesize. Therefore, on that local level, that particular cell does not make oxygen, is shielded from oxygen, and nitrogen fixation takes place on that cell. So, in the legumes, this is interesting. The bacteria that fix nitrogen in legume nodules make a hemoglobin-like protein called leg hemoglobin. Leg stands for legume here, and that, of course, has a great affinity for oxygen. So they make, just like our red blood cells have hemoglobin that has a high affinity for oxygen.
Elio, why is oxygen toxic to nitrogenase?
I don't know. Michael, I bet you know.
I think it just poisons the catalyst. I think remember, it's a redox enzyme, and I think the oxygen just sets the potential improperly, but I don't know the absolute reason.
You don't think maybe an allosteric thing? Oxygen is a little bit like nitrogen, O2 and 2.
It could be
It mistakes it for nitrogen, and it clogs up the works or something.
Well, according to Wikipedia, oxygen degradatively oxidizes the iron-sulfur cofactors.
That's right, of course.
So it's a redox thing.
Yeah.
It's a redox thing.
Yeah. Anyhow, it's interesting. There's a variety of strategies for shielding an organism from the oxygen it makes, in the case of photosynthesis, oxygenic photosynthesis, and in oxygen that's present in the atmosphere. So you have to shield nitrogen from it, and that becomes a very big deal. So all living things, other than bacteria and archaea, depend on them for fixing nitrogen, other than the man-made one. And it's not surprising that this leads to a lot of symbiotic relationships. We already mentioned some. The root nodules are a classic example of a symbiotic relationship between bacteria and a plant. And there are many other examples. But as I said earlier, keep in mind there are free-living nitrogen-fixing bacteria as well. I'd like to mention one interesting example, which is that of a fern called azolla. That's actually duckweed. You know, if you go to a lake that's kind of eutrophic, has a lot of phosphorus in it, you'll find that it's covered with little leaves. They look like little leaves, and they're called duckweed. I'm sure you know what I'm talking. All our
the entire Southeast is filled with those ponds and duckweed, and many homeowners association pay landscapers to kill the duckweed because it doesn't make their ponds look pretty. It clogs them up.
Well, it turns out that duckweed, which is known as by the name of azolla, that's the Latin name, is a fern. You never think of a fern floating on your lake, but there it is. A fern which has a symbiotic relationship with cyanobacteria. Anyhow, these so symbioses are common. So the question now becomes, we know about this mainly on land. How about at sea? What happens in the oceans where a powerful amount of life exists, as much as there is on land, and therefore needs a source of fixed nitrogen? And the answer is that some of that is, in fact, done in a symbiotic relationship. But, and this is known for higher plants, no question about it. But in the case of unicellular organisms, how do they do it? Do they have symbiotic relationships with, let's say, cyanobacteria, which are the most common photosynthesizers in the oceans and perhaps in the world? And the answer is yes. It's been known that diatoms have a symbiotic relationship with cyanobacteria. It's very interesting. I won't talk about it today, but this is an interesting subject. But the authors of 2 papers that I'm going to discuss have come up with an association which is different, and that is between microalgae. Diatoms are microalgae, but different kinds of microalgae, and cyanobacteria. So the one paper, the basic paper that I want to talk about, is by Thompson, Foster, Pke, Carter, Musat, Volo, Kuipers, and Zehr. And it's entitled Unicellular Cyanobacterium Symbiotic with a Single-Cell Eukaryotic Algae, and it appeared in 2012 in Science. And the second paper, I might as well mention it right now, is by some of the same authors, Thompson, Carter, Turk, Kubo, Malfatti, Azam, and Zehr. And it's entitled Genetic Diversity of the Unicellular Nitrogen-Fixing Cyanobacteria, UCYN-A, and its primnesiophyte host. Before you throw your hands up in the air, I'll tell you later what primnesiophytes are. The authors of this paper, the first paper is mainly by people from University of California, Santa Cruz, and the Max Planck Institute in Bremen, and a place in France. The second paper is some of the same people. It includes also Farouk Azam and Francesca Malfatti, who are at the Scripps Institute of Oceanography here in San Diego, and whom I know pretty well. So, what's the story? If you look at if you do this following experiment, you take water from the ocean and you put it through a cell sorter, flow cytometry. You find that you are, of course, going to notice a large number of particles, and these particles are mainly living things, and you can sort them by size into bacteria and something bigger. There's something bigger, which is called the picoplankton. Of course, it goes all the way up to whales, but I'm talking about the smallest of the eukaryotic components, called picoplankton. In other words, very small plankton. It's cells that can be as small as 1 micron across, up to 3 or 4, I think is the definition. By the way, the smallest eukaryotic cell belongs to this group. It's called Osteococcus tauri, and it is about 1 micron, 1.2, 1.5 microns across. It's an amazing example of micro-miniaturization, because in it there is 1 nucleus, 1 mitochondrion, 1 chloroplast, 1 peroxisome, and that's all there is room for, a little bit of Golgi. So it's gosh, I feel another digression coming. Is that okay?
Yeah, sure, no problem.
Okay, this has been the it's a flat, it's a round cell which is flat. It's like a disc, and it's thin enough that it can be studied by this wonderful new technique of electron cryotomography. Perhaps our readers know about it. It's a way of doing essentially CAT scans through a cell. and therefore reconstructing a cell which is in the frozen state and essentially still alive. It hasn't been fixed. And you can look through it and reconstruct it and essentially see what you would see if you could crawl inside of a bacterium. However, until recently at least, the technique was limited to very thin objects. So the only decent eukaryotic cells you could examine by electron cryotomography, this was done in Grant Jensen's lab at Caltech, was in fact Osteococcus tauri, and it's just wonderful. And maybe I'll put that in the show notes as well, because if you want to see what you would see if you could crawl inside a eukaryotic cell, this is your chance. Anyhow, now let's come back. Let's see if we can. We were doing flow cytometry, right?
That's right.
Remember that?
Yep.
Okay.
And the way we were measuring it was not with antibodies, but with PCR.
That's correct. That's quite a way of doing it, right?
It's a hard way of doing it. There's a lot of work in both these papers.
Yeah, indeed. But PCR, of course, lets you, depending on the probes that you use, lets you differentiate between the prokaryotes and the eukaryotes. And you can find, and that's the point of the paper, that a high proportion of the particles consist of a associated eukaryote and prokaryote. Okay? And if you look at the prokaryote, which has been studied, has a fancy name called UCYNA, and they give it also a Latin name, but I won't bore you with it because it's a candidatus. Candidatus refers to bacteria which cannot be cultivated.
And that's the U. That's what the U means. It means
that's right.
Uncultivated, and CYN is for us poor laymen, cyanobacteria.
Right.
So that's the way I kept them straight.
Okay, good for you. That's exactly right. Anyhow, the Cyanobacterium is known, as you just said, not cultivated. It has an interesting property, however, which called their attention. Namely, it has a reduced genome. I think it's, 1.6 million bases, something like that. Very reduced, much less than free-living organisms. And it's missing a whole lot of things. It's missing the photosynthesizing system 2, which is the oxygen evolving system in photosynthesis, takes care of oxygen. It lacks rubisco, said to be the most abundant or one of the most abundant enzymes on Earth, which is responsible for CO2 fixation. The tricarboxylic acid cycle, enzyme for the biosynthesis of amino acids and nucleotides. Therefore, it needs a source of organic carbon. It cannot fix carbon by itself. It may be an alga, but boy, it's not much of an alga in those terms.
And remember that when it loses photosystem 2, in photosynthesis, photosystem 2 is what provides the reducing power or the ability to fix carbon. The fact that it was missing rubisco is one thing, but the fact that it lost its ability to capture light and have that source of electrons where it evolves the oxygen, because it gets the free electrons that makes the reducing power, it effectively, for all intents and purposes, makes this microbe a heterotroph.
That's right, exactly. It is a heterotroph. It cannot live without a organic source of carbon, which called the attention of the authors. They said, well, if that's the case, where does it get its carbon? And a good possibility is that it lives in symbiotic relationships with somebody that can provide them that, namely an alga. And sure enough, this is really very nice work because it's done carefully. They realize that the association, they go later on, they look at these things. They can actually look at them under the microscope, and they see an association between a larger cell, the alga, and a smaller cell, this measly cyanobacterium. And they realize that if they shake the thing a little bit too vigorously, the two things come apart. And the shaking happens when you do the procedures that you often do to concentrate material from the ocean, where things are essentially in a dilute state, So you like to concentrate them before you do something. So you spin them, you do things like that. And when you centrifuge them, they come apart. But so they knew that they were there from the flow cytometry, but in order to retrieve them, they had to be careful. So what they did is to simply take seawater and filter it directly. And put it directly through the flow cytometer. That's dilute, you get much less of a signal, but it's good enough to do it. And when you do that, you find that they are nicely associated. And there is something like 60 to 90% of all the cyanobacteria are in association with a Pico Eukaryotic fraction. Okay, so now they find that the two things are connected, suggesting a close association, a symbiotic association. But because the connection is fragile, they don't think that these things are very tied together like many other symbionts. In bacteria, you have symbionts which are very closely tied to each other, bacteria and archaea, and they make filamentous connections between them. These guys don't. They're just sort of sitting one on top of the other, and if you shake them too vigorously, they're going to come apart.
Is there probably a function for that loose association?
That's a good question. Well, you can make one up. I mean, I don't think they know.
Yeah.
Coming and going is probably a good thing in some cases, but
just like humans.
Just like humans, right? They come and go. So so now we've identified the cyanobacteria. I'm going to come back to that in a minute, but let me first tell you about the Pico Eukaryotes. Who are they? Well, in the fractions that they retrieve, they're very diverse. There's all kinds. But the bacteria are primarily associated with these guys called, and forgive me again, Premnesiophytes.
Mm-hmm.
So who they? Well, it gets worse because I can tell you the names of some of the genera that are involved here, and this is a gorgeous one. Brarudosferia and Chrysochromolina. I'm telling you, no wonder some people swear off biology because it's a bunch of names, but never mind. Who are these Pico Eukaryotes? Well, there's unicellular small algae, which have calcium carbonate plates covering them. And the classic one, which is not one that we're talking about here, is Emiliana huxleyi. Remember that? We talked about.
Mm-hmm. Oh, yes.
Yeah. Yep.
If you remember, Emiliana huxleyi is one of the most gorgeous things in nature. It's covered by these plates, which are sort of radially arranged. It's just a very beautiful thing. Please Google coccolithophore, which is the name for these guys. They're called coccolithophore because these plates are called the coccoliths. And they are, if you Google them, you're going to get this is eye candy.
Mm-hmm.
Anyhow, they are the guys who make huge blooms, and their shells are deposited in such huge amounts that they made for the White Cliffs of Dover and other huge sources of limestone. Anyhow, but they're not it. These are two different ones. These, in fact, are just as gorgeous and a very strange shape. They make they have pentagonal plates on them, which are a pentagonal plate. If you take a pentagon and you want to make a 3D structure, you end up with a dodecahedron, a 12-sided structure, right? That's exactly what they look like. 12 sides of pentagons. And they're very distinctive. So they've identified them, and they're likely to be the host, if you wish, or the other partner in the symbiosis. So now the question is, can you show that this is actually what's happening? Can you show that there is, as you'd expect, an exchange of carbon from the alga to the bacterium? And while you're at it, wouldn't you want to also find out that the nitrogen flows from the fixed nitrogen from the bacterium to the algae? This would be a perfect symbiosis, tit for tat, right? So what do? They add heavy isotopic nitrogen in the form of N15 and heavy isotopic carbon in the form of C13 bicarbonate to a little sample, and they say they incubated in, quotes, in situ conditions. Now, that is, I think, called a microcosm, when you take a little bit of the volume of the ocean and just sequester it essentially in a test tube. But what this means is they took a test tube and filled it up with water.
Mm-hmm. and they shine some light on it.
And shine light on it, just as if it were in the ocean. And they may have run some air through it. I don't know, agitated a little bit. Anyhow, so now the question is, how do you find out what's happening? And this is a technique called in situ hybridization, nanometer scale, secondary ion mass spectrometry. Let's call it SIMS. Okay, these are some of these fancy techniques where you can do mass spec on a single cell, and if you can do mass spec, you can determine the ratio of C13 to C12 and N15 to N14, right? And sure enough, when you do that, you find that there is both heavy isotopes are found in both partners. That is, the bacteria have acquired C13 carbon, and the algae have acquired N15 nitrogen. So it looks good. It looks like this is the story. So we have direct evidence that this is a symbiotic relationship between two adjacent cells. We don't know what holds them together, but they are. And in this process, this is what happens. Now, think about it. This is a little bit like what could have happened early in life history. It is when a cell acquired a plastid, a chloroplast, which used to be a cyanobacterium. Could this be the initial stage of that process? What do you think?
Yeah, sure.
I think it also has profound implications when you consider the early Earth as oxygen levels began to rise out of the blue-green algae communities.
Cyanobacteria.
And you begin to consider the volcanic activity and the whole issue of asteroids hitting the planet and darkening, and the various photosystems that are absorbing light in different wavelengths. You could understand how this uncultured cyanobacterium may have had been selected for against losing its photosystem if it was unable to absorb that particular light wavelength, and then it hooked up by accident. And then this evolution of this symbiosis between the cyanobacterium that still retained its ability to fix nitrogen and the heterotroph effectively, or the autotroph feeding its excess carbon, because it was still able to absorb the right wavelength of light, while at the same time it lost its ability to fix nitrogen because it was evolving too much oxygen.
Right. Well, this, I think, is very appealing, and it's nice to think that you're looking at the throwback to an early stage in evolution, isn't it?
Well, this particular association is probably very ancient, right?
Well, they think so. Well, the algae, this particular algae, go back to the Cretaceous, which is, I think, what is that? 400 million years?
Yeah, 400 million.
Yeah, and so it's at least that ancient, and you would then, actually they propose that paleontologists or paleobotanists get busy and look for these things, because they can tell the algae, but they probably have never thought of looking for an associated bacterium in fossil sediment. So, this would be nice if somebody came up with a picture of that. But,
Elio, do we know if the sharing of these isotopes, does that require the cells to be attached, or do they do any experiments that try and disrupt them and see if you still get uptake of the isotopes in the same way?
That's a good question, but I bet you that I don't remember. I don't think they must have done it, but because they obviously know how to separate them. That's a necessary control. I don't know that. I don't remember them doing it. The dilution effect would be sufficient, though. I mean, the diffusion, even at a fairly short distance, would be enough to dilute out the product. So I would expect that this would be a negative control, all right?
Yeah, you could just vortex it, right? And
right, exactly.
Disrupt them, yeah.
Do the same experiment with the vortex material and show that there's no transfer. Let's pretend they did it.
Okay. Yeah, I'm looking. I don't find it. That's why I'm asking.
Yeah. That's nice. It would be a necessary control. But as I say, just the distance between the two partners would be increased so much that I expect it would get diluted out very rapidly. The amounts we're talking about are very small, and it takes very sensitive techniques to detect them. So I'm confident that this was okay.
They say that 95% of the fixed nitrogen is transferred from CYN to the partner, so it's a lot. It's pretty efficient.
That's right. It's very efficient. So you would expect a nice mechanism, because after all, these guys are three-dimensional. So you have a ball on top of a ball. It's not just going to make the nitrogen on the adjacent surface, and the surface is touching. It's going to spread out a little bit. So maybe there is some mechanism for transferring that's a little bit better than just diffusion.
Elio, there's no example of a relationship such as this where the bacterium is an endosymbiont, right?
Oh, yeah.
There are?
Well, the rhizobia are endosymbionts.
Okay.
Yeah. And remember, in the pure culture sense, there are cyanobacteria that differentiate and form a heterocyst where they don't
about that.
Where they don't turn on their photosystem.
No, but that's not what he means. He means a symbiosis. That's not an endosymbiont. That's the same bug. That's the same cell. So I don't know that there are other examples of endosymbionts with multicellular organisms.
But not unicellular as here, right?
That's right. This is the diatoms is the only case known, and they're not endosymbiotic. No.
They do say here in the discussion, it is still not known whether the cyanobacterium is an endosymbiont or lives on the surface. Yeah. However, the sensitivity to disruption and imaging indicates that it's a loose cell surface. So they must have done that experiment, because they say the sensitivity to disruption. So,
well, what they mean here is that if it were an endosymbiont, it couldn't disrupt it.
Correct.
It would be within the membrane of the eukaryotic cell.
Yeah.
So let me finish up with just giving you a sense of the second paper, because it doesn't add anything to speak of to mechanisms. But the second paper deals with the fact that you can, if you look all over the oceans of the world for these bacteria, you find that they divide conveniently into three clades, three ecotypes, if you wish. And they're found all over the place. They're found in the Bering Sea, in the Sea of Japan. Where all are they found? They're found in, the Sargasso Sea.
They have a map in their paper that's pretty impressive.
That's right. They're really all over the place. And in the open ocean, even the Mediterranean and so forth. So the three clades, three kinds, they're very closely related. They have a lot of similarity. They differ, interestingly, mainly in codon preference. That is, the third base of their codons tend to be different, which says that are not altered at all in amino acid sequence. So it's only when you look at their sequence that you find the difference. There's no functional difference in terms of these, if you wish, mutations. But this is useful to determine how they evolved. It's interesting to see how they spread. In other words, for ecologists, this is really an interesting study. And there's quite a few other papers on the subject. I just chose these from this particular groups, but there are other papers that deal with similar findings and confirm it in part. So I'm going to leave it at that, but I think that this is a good story in the making, and it's really surprising what you can do when you have good techniques. Nice work.
It's very cool. So just to put the cycle together, the fixed nitrogen eventually ends up back as nitrogen at some point, right?
That's right. That's right. In fact, I didn't say that, but the cycle of nitrogen in nature is pretty complicated, and there's lots to it. And someday we ought to talk about it and maybe give Michael a chance. Mike knows everything, so I'm sure he knows about that too.
Oh, I was tortured as a small graduate student, so I learned all about the nitrogen cycle. It's a wonderful thing.
Yeah, let's revisit that sometime.
And bacteria have a big role in that too.
Oh yeah. Oh boy. They're all over the place.
Well, bacteria have this big role in global mineral cycling in general. I mean, they're intimately involved with sulfur cycling. We all talk about the carbon cycle a lot, and the nitrogen cycle, as Elio started the story with his Talmudic question. It really is probably the most important aspect for life, is understanding how the nitrogen as a noble gas can be actually converted into something that can exchange electrons. And because the noble gases don't do anything.
Noble gas, is that really noble?
Now, your nitrogen's a noble gas.
It is. Huh?
It's noble.
Okay. I don't see it, but
my
okay.
periodic table. I gotta look.
You better, otherwise
I'll
you're
get
gonna
letters. I'll get letters. Noble gases. I'll look it up.
No, it's not. Helium, neon, argon, krypton, xenon, and radon.
I was off by one.
It's not a noble
I was
gas.
off by one column in the
forgiven.
periodic. I'm not a chemist today.
I'm sure people are already going nuts, Michael.
They're writing the letters now. Oh, God.
Here, let me give you Michael's email.
Yes, post it. Post it. It'll fill my Christmas email box.
Send them to him.
Send them to me, yes.
Thank you, Elio. That was lovely. I learned a lot. We have some email to answer, and the first one's from Kieran, who writes, I've just finished listening to TWiM 92, and it was interesting.
She's caught up.
As always. It is a pleasure to listen to all of you discuss these fascinating topics. At the end of the episode, you talk about probiotics because it was a question from a listener basically asking about the efficacy of these. I would very much like to hear a probiotic-themed episode, as my research thesis area is in some way related to the probiotic-prebiotic area. The two big names that come to mind are Todd Klenhammer, North Carolina State University, and Willem de Vos from Wageningen University in the Netherlands. Both are well known in the area of lactic acid bacteria research, which for obvious reasons is very intertwined with the probiotic research area. Anyway, I just thought I'd mention these names, and thank you and your co-hosts again for the great work that you do for making microbiology so accessible to everyone and for teaching me about the great diversity that exists beyond my own thesis area. Keep up the good work. P.S. I also like Elio's Small Things Considered Snippets idea.
Oh, good.
Okay.
Good. We'll do some more of that.
Kieran is a PhD candidate at the Cork Institute of Technology. That's in Ireland.
It is.
Cork.
I've been to Cork. Lovely place.
Really?
Very interesting.
Huh. Yeah, we will do a probiotic episode. We'll work on it next year, Kieran. Good idea. Next one's from Adam. My understanding is that each of the two existing polio vaccines, OPV and IPV, each have their own issues which might prevent them successfully eradicating polio. To summarize the issues, 1, because OPV replicates in the GI tract, it can mutate and revert to the wild-type pathogenic strain. This is correct. 2, IPV is delivered by IM injection. It protects against paralysis but does not sufficiently inoculate the gut. As a result, people vaccinated with IPV can still be infected with and shed wild-type polio. Correct. Adam, we talked about this.
Doesn't do anything to the gut. It's dead.
That's right.
It's dead.
We did talk about this, Adam, I think in the last episode, right?
You did. That's the way you finished it off.
You have had many great discussions about these issues and many debates about whether either vaccine could be used to eradicate polio. Theoretically, immunizing with OPV would come very close to complete eradication, but would be stymied by the rare revertants. While there seems to be great uncertainty whether either vaccine could do the job, it does leave open another possibility. Shouldn't polio eradication be possible if both vaccines are given to everyone? If IPV was given first, and then a few months later OPV was given to every individual, that would seem to solve the problem. IPV would prevent paralysis, and a subsequent vaccination with OPV would provide gut immunity. In this scenario, there would still be rare cases of people mutating OPV into the wild-type virus, but those people would not be at risk of paralysis because outside of the GI tract, they would be protected by their prior IPV immunization. Eventually, no one would be at risk of paralysis because of IPV, and no one would be able to carry or shed the virus because of OPV. At that point, immunization could cease. There are, of course, plenty of logistical issues with doing this. It would substantially increase the cost of immunization and makes the job of distributing the vaccine regimen even more challenging, especially in areas embroiled in conflict. This means economic and political issues may prevent this approach from being successful, but hopefully it would solve the theoretical issue with either vaccine being used individually. I apologize if you have already discussed this possibility. I try to catch everything, but occasionally I'm forced to focus on what I am actually doing while I listen. Yeah, well, that's a tough one. We touched on a little bit of this. You're absolutely right.
Let me ask you, Vincent, if you give the IPD, will the oral vaccine catch? Will the live vaccine, aren't you immune?
No, because the OPV is taken by ingestion, so the IPV is not affecting that at all.
Oh, that's right. Of course.
And then when the OPV gets into your bloodstream, it just does a little booster effect, and you're fine. So that was actually used for a while in the U.S. around 2000, and it is actually a strategy that is suggested by WHO in countries where you're concerned about vaccine-associated revertants. However, this is not the overall strategy that WHO is recommending. They're recommending, since most of the world is now being immunized with OPV, they want to now transition to IPV. And it's been found very recently, we discussed this last time, when you take someone who's been immunized with OPV and then you give them a shot of IPV, it boosts their gut immunity unexpectedly. But this is a good thing because now you can go in with IPV and immunize and boost gut immunity, and you won't get this circulation that's a problem, and you won't have vaccine-associated revertants. So you're correct that both vaccines have a place in the eradication plan, but for the most part, it's going to be OPV, IPV.
And what you're actually suggesting is a new vaccine in reality, because if you're going to give the oral and the injected at the same time, you really have to change the question that you're asking. And if you're trying to make the argument for global eradication, it's going to require study to see which strategy will work best, or do we now already have enough data to make the argument that the WHO strategy is sufficient to result in global eradication? Because we did eliminate polio from the Western Hemisphere.
That's right, using OPV.
Yeah.
So just to clarify, they won't be given at the same time. They will be given sequentially, right? They will.
And that's what I've said.
But it's the question. It's I'm lumping
of course
both into one vaccine, because that's how it will likely be paid for by the WHO or these governments, because you will call it one vaccine in two parts.
Right. I think it's a good question. It's not certain. You're right. The only way in a few areas where we have eradicated polio is using OPV. So who knows? But this is in addition to OPV, so I don't think it could get worse. So I think it would probably work. The real issue here is injection and cost. IPV is injected, so now you have to have people who know how to use needles. And then you have to pay. Right now it's more expensive, and hopefully the cost will drop when you make a lot of it, but that's a concern as well. Very interesting issue, though. All right, we have two more. One is from Ken. I had an interesting thought today while reading about problems with antibiotic usage. I'm hoping some sort of answer is provided so that you can tell me I am a nut job, or this is an interesting idea, or somewhere in between, please. Okay, we'll do that. There is certainly a lot of study as to what compromises the microbiome. This has led to ideas of weight control, mood moderation, and other health stasis attributes. One thing that has always bothered me has been looking for the, quote, safe, unquote, microbes, and not necessarily the balance. My thought was, could people that have side effects from antibiotics actually identify a risk factor for items such as C. diff or even some cancers? The idea being that if a person's balance at an earlier date has a normal population ratio, that increases the risk of the afflictions later in life. This could be something to allow for earlier screening or modify future treatments for other ailments in the future. It seems like this idea is wide open. obviously the idea shares some aspect of using flatulence as a marker as well. Maybe a gas spectrometer measurement could provide a quick and easier route for treatment. I got a vision here.
Elio laughs, but gas chromatography has been used by anaerobic microbiologists to speciate who's there.
Yeah, it makes perfect sense, right?
That's the famous work of David White.
Of course, that seems a lot more varied with a lot more problems than idea of antibiotic side effects. The latter seems like a simple piece of medical history that could be readily studied. What do you think, Michael?
I think this individual has described a potential roadmap for the NIH of, how do you, there are many questions he has placed in this very simple email. And I think all of these are worthy of study, but how do you begin to define the question? Because you have to define your population. You have to define the question you want to answer. I mean, the whole issue of flatulence while being funny, that is an easy way. But how do you collect a gas sample? Do you feed everyone beans in the waiting room and hope? Will that bias the measurement because you're feeding them a particular carbon source? All of these things, and it goes back to the earlier email about looking at probiotics, and maybe that's the way to do the study is give patients a probiotic regimen for a defined period and then have them come in and measure their community and then allow them to wash out of the probiotic and then have them come in and measure their community again to understand what's going on. The issue as to weight control, mood moderation, and the other health status attributes, we just don't know enough about the microbes. It's easy to speciate with 16S, but we really don't know about the metabolic products and how they interact with our immune system, because there's a lot of crosstalk, as we did a few papers ago, the crosstalk of the bacteria talking to simple things like worms. We know the bacteria are talking to our neuroendocrine system as well. And I go back to that seminal ASM where I first fell into the meeting on microbial endocrinology, and that's really microbes in our endocrine system.
So Michael, just doing the microbiome isn't enough then. You have to look at the metabolome, right?
Right. Absolutely. And that's where folks are going.
So in a lot of studies now where people have simply been doing 16S, they're going to add metabolome to that, I would presume, right? Right.
And, they're freezing a lot of samples, and the 16S is real easy to do with these tags that they're putting onto things. Mm-hmm. But also people are doing shotgun sequencing of everything that's in the gut and assembling that. So as our understanding of other things that as our sequencing gets more, as our databases become more robust, we can go back to those old libraries and begin to characterize using new tags and new probes to see what was there. You can effectively re-look at the data.
That brings up a point. It bothers me that when you say everything is in the gut, it's likely to be feces they're looking at.
Yes, and that's wrong too.
This is a paradox. The easiest biological sample to obtain from a human is feces. The hardest biological sample to obtain is what's really in the gut.
Yeah.
Because if you go to somebody gets operated, he's been cleaned up. If you look, where are you going to find somebody? How are you going to go and look for the bugs which are actually in the gut? And the gut is not just a tube. Therefore, I think that a lot of the microbiome suffers from this. In animals, you can do something about it. In people, it's extraordinarily difficult, and we shouldn't confuse looking at the contents of the gut with looking at at what's in feces. It's not the same thing.
Oh no!
People are aware of this. I mean, I'm not saying anything that people don't know, but it's easy to confuse the two things. Maybe we should deal with this one of these days. Maybe we should bring somebody who can discuss this rationally. It's a big deal. I mean, all this intestinal microbiome, there are thousands of studies now, and they all deal with feces.
I agree.
But not all.
And the problem is that we've seen a few interesting results just using fecal microbiome, and that kind of convinces people, well, yeah, this is fine to look at, but you have to look in the actual mucosa. And then the problem is the intestine is very long, and I bet it differs every foot.
I bet you it differs. Remember, these microbes are small, and there are, when you discuss, and I always butcher the French, the terroir of wineries, of how you grow the grapes. There, I am certain, is a terroir of our gut based on how you move through. And we did that paper early on in TWiM, looking at the South Africans who were using traditionally farmed vineyards and then organically farmed vineyards and measuring them in between. And they were seeing differences in the communities. And it's all about sampling. And I think Elio brings up a seminal point. It's where the sample is coming from. And in the case of the winery paper, they were going to the dirt and actually pulling out the dirt and extracting the microbes. They weren't like flooding the dirt with water and then seeing what comes downstream, which is effectively what we're doing with stool.
Yeah, that's right. No, it's a good point. Absolutely. All right. One more from Harold, who writes, Vincent and friends, longtime TWiV listener, used to listen to TWiM when it was on the TWiT network. I don't remember, Harold, it ever being on the TWiT network, This Week in Tech. I'm sorry. Maybe you're thinking about Futures in Biotech. Temperature in C, 78.F. Wait, temp in cubes. Oh, in your cube, 78.8 F. It is always bright and fluorescent in the cube farm. I get it. It works in a cubicle. As a lifelong high BMI obese person, I'm always looking for research on obesity treatments. I saw this study and wanted your comments. And he sent a link to a Cell paper called Human Genetics Shape the Gut Microbiome. And basically, they did a study in the UK looking at 416 twin pairs, and they identified many microbial taxa whose abundances were influenced by host genetics. And they cultured some of this and transplanted it into germ-free mice and showed that the mice would get obese, which is something that has been done before, and we've talked about that here on TWiM. And he continues, can transplanting the gut microbes of low BMI people into high BMI people lead to weight loss? Will this be a new angle for weight loss scams? What kind of diseases could you transmit using gut microbe transplants? So if I remember, Michael, putting low BMI into high doesn't do that, right? In mice anyway.
No, it's the other way around. The high BMI into a low BMI mouse resulted in a fat animal. And he's asking the question the FDA is currently addressing, how to qualify donors for these stool transplant pills that are being made. And the FDA is actually phrasing those questions of what tests do you have to look for, what parasites do you have to screen for, what aside from the pathogens, because we don't know if there are good microbes that are actually bad microbes in certain genetic types, as this Cell paper is potentially suggesting.
So what bacterial infections would you be concerned about transmitting by a fecal transplant if you didn't even check the donor, for example? A C. diff, right? C.
diff is the first and foremost. The enteroinvasive E. coli that may be salmonella, and because some people generally salmonella is always considered a frank pathogen, but there are carriers of everything.
How about cholera?
Cholera, not so much.
How
Cholera
about
definitely.
Campylobacter?
Yeah,
Campylobacter and cholera. Remember, that's how cholera got into Haiti. The soldiers brought it in, and they deposited it through the latrines, and it made it into the water table. So I think certainly those things, and let's not forget Typhoid Mary.
Yeah, sure.
I mean, we have that from the turn of the 19th
viruses?
to the 20th century.
I was just going to say there are
viruses.
There are plenty of viruses there. The noroviruses, the rotas, HIV is in there. So you really have to check for a lot of things.
And that's not inexpensive. And
wouldn't it be cheaper to just make an artificial mixture?
I think so. Yeah. But
well, that's also what a lot of companies are beginning to explore. About 15 years ago, when I was working on whole plant corn silage, making inocula, that's effectively what we were doing. Is we were trying to develop an inoculum that would prepare ensiled forage for animals, so they would gain more weight with less food. And we worked on the early microbiome of whole plant corn silage.
So weight loss scams, I could see that. Of course, I mean, I think probiotics to a certain extent are hyped a lot, and they don't do a lot, right?
Correct.
Although we have to get someone on here to talk about that, but I can see companies. But you can't sell feces in capsules. The FDA is not going to let that go over the counter. So I have to do something else.
Not yet. But no one's going to call that a health food or a food supplement.
No.
Well, you never know.
That'd be the way around it, right? Call it a food supplement.
Yeah, you call it a supplement.
All right, that'll do it for TWiM 94, and you will find this and all the other episodes for your sampling at iTunes and also microbeworld.org/TWiM. And we love answering your comments and questions here on the show. And if you have something you'd like to know about, send it to twim@twiv.tv. Elio Schaechter is at Small Things Considered. You can find that online. Thank you, Elio.
Oh, my pleasure, of course.
Very nice story about nitrogen. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
Always a pleasure. And I am Vincent Racaniello. You can find me at my blog, virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.